So, today we're diving into something working at, like, the absolute speed limit of biology. We're talking pumps cycling thousands of times a second.
Over 5,000 times a second. It's incredible.
But here's the really stunning part, the sort of counterintuitive thing from this paper we looked at. The insane speed of these specific pumps, the PMCA2s, it doesn't just come from the protein itself. It critically needs a simple fat molecule in the membrane.
Yeah, a phospholipid PIP2. It acts like a kind of structural latch almost to enable that speed. It's fascinating.
Okay, so maybe let's back up just a tiny bit. These are plasma membrane calcium ATPases, right? PMCAs.
Exactly. And their job is absolutely vital. Think about processes that need super precise timing, like neurons firing in your brain. PMCA pumps have to clear out calcium signals almost instantly to reset the cell. If they're slow, everything gets messed up.
Right, because calcium signals control so much, and you compare these to other related pumps like Circa, the speed difference is just wild.
Oh, absolutely massive. Circa pumps calcium into internal stores, maybe cycles a few hundred times a second, tops. PMCAs, they need to be in the kilohertz range, thousands of cycles per second.
So the big question the researchers tackled is how? How does PMCA2 hit these almost unbelievable speeds?
Well, the first clue was really in the basic design, the architecture. They used cryo-EM, so freezing the pump and imaging it to see its structure at different points in its cycle. They mapped out 8 different stages.
8 stages. And comparing it to the slower Circa pump revealed something pretty fundamental about its movement.
Totally. So in Circa, there's this part called the end domain. During its cycle, it rotates like a huge amount, 66.5 degrees.
Okay, like swinging a big lever.
Exactly. But in PMCA2, that same domain, it barely moves,
Yep.
just 8.3 degrees.
Oh, that's a tiny nudge in comparison.
A tiny nudge. And think about what that means for energy. Less physical movement means less energy needed for each pump cycle. It's inherently built for low effort, high repetition work. That's the foundation for its speed.
Okay, that makes sense. Smaller movements, faster cycles. Now, the structures also showed another protein nearby, neuroplastin and PTN. And people had thought, maybe that's the secret speed booster.
Yeah, that was a reasonable hypothesis. It sits right there, like an accessory subunit. But they tested it directly using functional recordings in cells, and the result was clear. Neuroplastin doesn't actually speed up the pump itself.
Oh. So what does it do then?
Its main role seems to be, well, it's more about stability and logistics. It helps stabilize the pump structure and make sure it gets trafficked correctly to the cell surface, the plasma membrane.
So it's like the delivery truck, ensuring the fast machine gets to the right factory, but
Yeah.
it's not pressing the accelerator.
Precisely. Good analogy.
Okay, so if the protein's core design is inherently fast due to minimal movement, and neuroplastin isn't the turbo boost, what interaction, what specific molecule really pushes this thing into that kilohertz range? This leads us to that lipid, right?
Yeah. Exactly. This is where PIP2 comes in. The researchers had a strong suspicion about it. They did this neat experiment, measure the pump activity in intact cells, then use a special enzyme, CEVSP.
What does that do?
It's a phosphatase, so it specifically targets and basically chews up PIP2 lipids in the membrane.
Ah, okay, so they removed the PIP2.
And boom, the pump activity just plummeted. It showed that PIP2 is absolutely required for that top-end kilohertz speed. It's obligatory.
Wait a second. So PIP2 is needed for speed, but the pump's job is to get rid of calcium. Doesn't PIP2 bind when calcium is attached? How does holding it help it release faster? That sounds backwards.
That's the really clever part. It acts like a latch, but maybe not in the way you first think. When the pump is in its calcium-capturing state, PIP2 fits snugly into this specific pocket near where the calcium binds, and by binding there, it stabilizes a key amino acid residue, N841. This stabilization is what actually ensures the pump grabs calcium with really high affinity.
Okay, so PIP2 helps it grab calcium really tightly at first, like setting a trap.
It's exactly like setting a trap. But then, the pump needs to transition to release the calcium outside the cell. It changes shape.
Yeah.
And in that process, the interaction with PIP2 is broken. It pops off, essentially.
Ah.
And when PIP2 leaves, that residue is stabilizing, N841, it moves. And that movement nudges another critical residue, D873, the one actually coordinating the calcium ion.
And that nudge is the trigger.
That's the chemical trigger. The nudge changes the chemical environment around D873 just enough to shift its pKa.
Okay, pKa, that's acidity, basically.
Yeah.
How readily it holds onto a proton versus the calcium.
Precisely. The pKa shifts from about 6.0 to 7.0. So suddenly, D873 dramatically loses its affinity for the positively charged calcium ion and gains a much higher affinity for a proton. which acts as the counterion that gets transported back in.
Wow, so PIP2 holds it tight, then letting go of PIP2 causes a chemical switch that instantly makes the site repel calcium and grab a proton instead.
It's an incredibly efficient molecular switch. The removal of PIP2 initiates this rapid cascade, forcing calcium ejection. It means PIP2 isn't just like passively helping, it's actively setting the rate.
That changes how we think about membrane lipids, doesn't it? They're not just the environment, they're part of the machine's timing mechanism.
Totally. Critical rate-setting elements.
So, thinking about innovation implications. If PIP2 binding is so crucial for speed, and there's a specific pocket, is that a potential drug target?
Absolutely. And they provided proof of principle for that. They took a known inhibitor, sapsigargin, which usually targets SERCA.
Right, the slower pump.
Yeah. They found sapsigargin actually does inhibit PMCA2 as well, with a decent potency in IC50 of 2.6 micromolar. And critically, their data suggests it works by competing directly with PIP2 for that binding site.
That's really significant. It demonstrates you can target this PIP2 site with small molecules to control how fast calcium gets cleared from the cell, which could be huge for, say, neurological conditions or even certain types of hearing loss linked to calcium handling problems.
Exactly. It opens up a new avenue for drug development.
Okay, so let's wrap this up. If you had to boil it down, what are the, three main takeaways from this deep dive?
Okay, first, PMCA gets its incredible speed, partly from its structure, those really small, energy-efficient movements compared to other pumps. Minimal conformational change.
Right. Less effort per cycle.
Second, that membrane lipid, PIP2, is absolutely essential for hitting top speed. It acts as this chemical latch mechanism, enabling fast calcium release through that clever PMCA shift.
The latch and the chemical switch. Got it. And
third, because PIP2 binding is so critical and happens at a specific site, that site represents a completely new and apparently effective target for drugs designed to modulate calcium clearance.
a new druggable target. Fantastic.
So here's a final thought to leave you with. Since this whole ultrafast mechanism hinges on a specific lipid in the membrane, what happens if that membrane composition changes subtly? Not necessarily due to disease, but maybe just, diet or aging. Could those subtle lipid shifts tweak the speed of these critical pumps, influencing high-frequency signaling in our brains or hearts in ways we haven't even considered?
That's a really interesting question. How the cellular environment directly tunes these nanomachines. Lots to think about there.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.